introduce our speaker for today. We're very lucky to have Rachel Poretsky. She hails from the great urban microbial hotspot of Brooklyn, New York. She obtained a BS in biology from Brandeis University in 1999, PhD in marine sciences from the University of Georgia in 2008. She was a postdoc at Caltech in geology and planetary sciences and at Georgia Tech in environmental engineering. And she has done pioneering work looking at genes and gene expression of microbial communities in the open ocean, freshwater lakes and rivers, and wastewater, urban and engineered environments. And she is currently an associate professor in ecology and evolutionary biology at my first college, University of Illinois at Chicago, where she has been since January of 2013. Without further ado, I present Rachel Poretsky.
Hi, thanks. My mom just joined. Hi, Mom.
Hey, that's wonderful.
So she can find out what I do. So I'm going to go ahead and share. I have slides, and I will go ahead and share my screen. And you all can post in the chat if you have questions, and I think Charles is going to monitor it and let me know. But I'm happy to be interrupted. So yeah, I'm here to talk about microbes and why they're so amazing. And so I started out with this quote that was in this publication that came out already 10 years ago, but still relevant, saying how in the last 20 years, there's been these major technological advances, and we've learned so much about microbes in soil, in the ocean, and even the human body. And what we've really learned is that there's a lot that we still don't know. So we've learned that there's a lot of mysteries and a lot of things to yet be discovered. And I like this quote because it says, how can we even characterize all that diversity, and how did evolution generate this diversity to begin with? And this quote at the very top says, what we can learn from the microbes that sustain our planet and our bodies stretches the limits of the imagination. And this is why I love this field so much, and why I dedicated my career to this. So to start out with, some of you probably know, and some may know really well, but just so that we all are on the same page, what are microorganisms, also called microbes? So they're really things that are micro, that are tiny, things that organisms that you can't see with your naked eye. Usually they're a single cell or sometimes clusters of cells. And the major group, these are all major groups of microbes: bacteria, which most of you have probably heard of, archaea, which maybe some of you have heard of, but they're actually a whole other domain of life that looks a lot like bacteria, but they're actually more closely related to us, to these organisms that are eukaryotic, so things that have nuclei. Fungi, algae, protozoa, and then I put viruses in parentheses because we all know a lot more than we ever thought we would know about viruses, unless we've dedicated our careers to studying HIV, right? But viruses also I put in parentheses because they're not really living things. Like, we don't even know how to define if they're alive, because they only replicate in a host or with a host. So all sorts of microbes, and they're everywhere. What kind of unifies them? They're small, right? Micro by definition. They were first discovered with this is a picture of the very first microscope that Anton von Leeuwenhoek, he put pond water and he drew what he saw, and he was like, and these are even like paramecium, like things that are bigger than bacteria, but he saw these tiny things and he was like, whoa, tiny things. So microbes are small, but they're really abundant. And so here's some fun facts to put in your back pocket to share. They inhabit every environment that supports life. So anywhere that living things could be, there are microbes there. Sometimes there's not very many, sometimes they're really unique and really suited to a particular environment. Sometimes like a soil or the human gut, there's tons of different ones, but everywhere you can find life, there are microbes. There are more cells, microbial cells on Earth than there are stars in the universe. So it's estimated that there's like 2 times 10 to the 30th microbial cells. That's a lot on Earth. About 60% of the biomass on Earth is microbial. So that's more like just alive mass than plants, animals, anything else on the planet. If you were to take a teaspoon of seawater from the ocean, there's in that 1 teaspoon, there's millions of bacteria and probably 10 times as many viruses as there are bacteria. So there's lots of viruses that aren't as notorious as SARS-CoV-2, right? But lots of viruses that are also essential for maintaining the balance of life on Earth. So I like showing this because it sort of puts that in perspective. Any idea which one represents, which one is a picture of stars in the sky and which one is a picture of bacteria under the microscope? So I just love that they look so similar. So the top one is microbes, mostly bacteria. And you can stain it and see their DNA. You don't know who's who, but you can just see that there's the mass abundance of bacteria in a sample. This is a seawater sample. But really puts it in perspective. So top one, bacteria, bottom one is the night sky. Another fun fact, so I told you about water, seawater, a gram of soil has even more, so billions instead of millions of bacterial cells in a gram of soil. To put that in perspective, that gram of soil has more bacterial cells than the world population of humans. And this is a phylogenetic tree, which I put up here because this was published a couple of years ago showing all of life. So this represents all of life on Earth. And all of this top part, this is a way to look at how organisms are related to each other. All of this top part is bacteria. All of this purple part is bacteria that we don't know hardly anything about. This little part here, colorful part at the bottom, is archaea, this other microbial group that's pretty closely related or more closely related to us than bacteria. And then this tiny green sliver here is all eukaryotes. So that means animals, fungi, slime molds, plants, like everything else is in this tiny sliver, including us, of course. So again, most of the world is microbial. Dr. Marty Blazer, who's in New York, wrote a book called Missing Microbes, and he had this quote in there that said, humanity is just a speck in the massively bacterial world. We need to get used to that idea. So bacteria come in lots of shapes, sizes, really some really beautiful, some I think a lot of these are beautiful, but some really these are tiny, almost like tiny plants in the ocean that form these shells, and they're really pretty. Lots of shapes. Some form these branching structures. Some are spirally. Lots of different shapes, lots of different functions. We're intimately familiar with the bad ones, the ones that make us sick, give us rashes, are carried by things like mosquitoes or water that's not treated well. but they have a lot of good functions. And in fact, we are really concerned with the bad ones because they make us sick. But the good stuff they do is really important to both our health and the health of the planet. So things like fixing nitrogen, so providing nitrogen to leguminous plants, soybeans, and the like. All of that happens by bacteria. Fixing carbon. So the oxygen that we breathe, most of the oxygen that we breathe is produced by photosynthetic microorganisms in the ocean. So about half to a little bit more of the oxygen that we breathe is made by photosynthetic microbes versus trees and green plants that we see outside. So fixing carbon in other weird ways at the bottom of the ocean, really important to us and really important to the planet. And animal nutrition. So termites can't digest wood. Hopefully nobody has to deal with termites eating their house, but they eat wood, and they can't digest the wood. They rely on the microbes in their guts to break down that material. There's really cool associations with insects in the hindgut of cows. There's a cow like this at the vet school when I was at Georgia. They insert these fistulas, these ports in the side of cows, so you can actually reach me. I don't do this, but people who study the rumen fluid can stick their arm in these holes and take samples of rumen fluid. So yeah, animal nutrition, microbes are really important. Also, biotechnological applications, things like if you like soy sauce or cheese or fermented foods, alcoholic beverages, microbes are really important. Making possible sources of new fuels, treating wastewater. All of this stuff is really important to our lives, whether we think about it all the time or not. And it's mediated by microorganisms. And then, of course, the human microbiome, which has gotten a lot of attention in the last decade or so, all the tiny habitats on and in us that are full of organisms. And in fact, if you were to take off all the microbes on and in you, they would weigh about 2.5 to 5 pounds. So if you are concerned about your weight, you can be like, well, 5 pounds of that is microorganisms. But they're so abundant on us. There are about as many microbial cells on and in us as there are human cells. But if you were to count their genes, we're made up of a lot more microbial genes than we are human genes. So it's pretty incredible. I don't know if you can see my shirt, but it's mainly microbe because essentially that's what we are, is a habitat for microorganisms. Microbes also have a lot of unusual functions and can live in habitats that we find extreme. For them, that's just where they live. So places with really high salt, hot temperatures, lots of sulfide, really high pH. So I've worked here. This is in Mono Lake in California. Really high pH, really cold environments. Places where no other organisms can live, you can find microbes there usually. And like I started out saying, most are still unknown. So in that same report, they showed this this little toy boat represents what we know about microbes on Earth, and this big ship represents the unknown. It's a little bit, we know a little bit more than we did 10 years ago, but there's still a lot of mystery of the microbial world. So how do we study them, and how do I study microbes? In the environment is we actually don't look at, generally, we don't look at the organisms themselves, but we look at their DNA or their RNA, their genetic material. So we can go into these different environments and we can extract their genetic material and then understand the who's there, what microbes are there, do like a census of all the microbes in the environment and find out what sorts of functions they're either capable of or that they're doing. So I wanted to show you some microbial ecology. So this is my field, studying microbes in the world, in the environment. A little sense of where I've done this. So actually this first picture is the RV Kilo Moana. It's a research vessel out of the University of Hawaii. And so I've done work in the North Pacific on this ship. I've also done work on the South Pacific, on this same ship. And that was a cruise in the South Pacific that went from Australia to Fiji. And that was almost 2 months on this research vessel. Here I am with one of my colleagues working on or cocking the water, the Niskin bottles, they're called, but the sample bottles that we collect seawater in. And interest or fun fact about this cruise was it was about 2 months, and I was actually at sea during Passover, and it was my first time missing Passover at home or with family. And so I actually brought matzah and a haggadah and some other, I don't know what, along with all our scientific equipment. I brought some Passover things on the ship with me, and we had an afikoman hunt on the boat, and it turned out that one of the science technicians on the ship. So usually there's the crew, there's a science technician, and then there's a team of scientists. And so one of the science technicians on this cruise was Jewish, and he was like not a very practicing Jewish person, but he was so excited, and he ended up calling his grandmother from sea and telling her that he celebrated Passover in the middle of the ocean and made her month probably. So I've also been on a research vessel that has this deep sea submersible, Alvin. This is a picture from Alvin where you can go to the bottom of the ocean. And we were working on the Costa Rican margin, looking at the microbes that live in the sediment there. This is Mono Lake again, this really high pH soda lake in California. This is Sapelo Island, where I did a lot of my PhD work off the coast of Georgia. Where else? Yeah, this is in the Gulf of Mexico, looking at sediments. Same sort of sampling devices. Yeah, so lots of exciting work in lots of exciting places. And so I kind of wanted to spend the next half, I don't know where I am with time, but telling you a little bit about what I'm doing right now, and what people I'm working with are doing right now, and some of the exciting developments. So this is a former postdoc in my lab, Melissa Pierce, and another student in my lab, Emily. So they were involved in microbiome work related to these sablefish. So this is a project I had with a NOAA fisheries lab, or have still. Melissa and Emily both left. Emily graduated, Melissa got a job. But we're still working with this project with the Fisheries Science Center in Washington State. Looking at the development of fish and the role that the microbes in the water play in fish growth and development. Another former student, now postdoc with me, this is Jared Hampton Marcel, works a lot on human microbiome work, and his work focused on swimmers and how exercise can change your microbiome, or how your microbiome can influence your exercise training and volume. And so he works with me, and then somebody in the kinesiology, this is Craig Horswell in kinesiology, who knows more about humans and sports than I do, or exercise, I should say, exercise science. And looking at how our microbes are intimately involved in human function and health and ability to exercise and adapt to changes. And then so a lot of my questions from the start of when I've been from the start of this work have been related to how microbes play a role in transforming carbon. So I talked a little bit about carbon fixation, like photosynthesis, the same idea of taking carbon into biomass or using carbon, respiring, breathing. And so we've been working for a number of years in Lake Michigan, also on research vessels, not quite as large as the marine vessels or oceanographic vessels, but research vessels out in the Great Lakes to look at how microbes are influenced by changing carbon dynamics. Maybe some of you have heard about issues in Lake Michigan with invasive mussels, and the mussels have drawn down the phytoplankton, the tiny photosynthetic organisms that are in the water. And those photosynthetic organisms provide carbon to everything else in the lake, so higher trophic levels, including fish. So we're interested in studying how those carbon dynamics have changed the overall health of the lake, and how that changes from near shore to offshore. And similar questions on carbon dynamics. We've worked in Alaska, in the northern tundra, in Alaska, looking at carbon dynamics in the soils there, because the snowpack will provide this insulating layer, and with reduced snowpack, as temperatures increase, that changes how carbon gets stored and released from soils. So maybe some of you are thinking about how this all kind of ties together. Like, what the heck does understanding the athlete microbiome have to do with marine science or understanding freshwater microbes? And the simplest answer I have for you is poop, which I like to say that I have transitioned from working heavily on in these exotic locations like the Costa Rican margin and Fiji, to working a lot with poop related work. But it's all connected, because I tell my students too that one of the reasons that microbes are so fundamental to the planet is because they work together. Some produce something that another set of organisms will use. So waste products kind of unite everything. That all the biogeochemical cycles and all the interactions on the planet is all intertwined. Like, you can think about how photosynthesis releases oxygen that we then use. So that's a really concrete example. Or we release CO2 that plants can use for photosynthesis. That's a really concrete example of waste of one organism being food or nutrients for another. And so this really drives my work. Like, how do these interactions happen, and what happens when that changes, and what happens when there's some external change? How do organisms respond? So here I've in Chicago, I've really done a lot more urban microbiology. in both Lake Michigan that I mentioned, but also in the Chicago River related to wastewater treatment plants in the area. And this is my former technician and a grad student sampling up at the north end of the Chicago River, right by, it's all white here. This is kind of rapids that come out of the wastewater treatment plant up there. So Chicago has 3 main wastewater treatment plants that service our area, the Chicagoland area, 3 big ones. And they all 3 release their treated effluent. So whatever, they treat the wastewater and they release what comes out into the Chicago River. We also have here in Chicago these combined sewer overflows. Fun fact, you can sign up to get text alerts whenever there's a CSO event from the Metropolitan Water Reclamation District. Anybody can do it. You can go to their website. But what it means is that when it rains a lot, stormwater mixes with wastewater and before it gets treated, and sometimes more often than we would like, untreated wastewater gets released into our waterways. And this is kind of not unique to Chicago. It's a feature of many big cities, these combined, big and old cities, these combined sewer overflows. But it's a problem enough here that this was right a year or so before I moved to the area. The EPA fined the city of Chicago, maybe some of you remember this, fined the city of Chicago a lot of money and said that the Chicago River has to comply with recreational water standards. So prior to this, the river it did not have to, because it has a really interesting history and was built as this sewage and shipping channel. And because of that, it didn't have to comply with recreational water quality standards. But many of you have probably seen, or some of you may have kayaked or and been on the river. And so the federal government said, this is not just a sewage and shipping channel. People use this for recreation, so it's got to be cleaned up. I thought it was kind of funny. This was a little bit after I moved here. There was this headline, the new water disinfection plant helped Chicago join the civilized world. And so what happened was, when the federal government issued this mandate, they said, so at the time, Chicago was the largest municipality in the country, probably in the world, to treat our wastewater, but not disinfect it before releasing it. And so when the federal government said you have to, they, in 2 of the plants, the one up north and the one on the south end, both had to build disinfection capabilities to disinfect wastewater. Unfortunately, Stickney, which is the one that serves Oak Park and much of the near west suburbs and the kind of all the center of Chicago, it's the largest, maybe the largest wastewater treatment plant in the world. It serves about a little under 2.5 million people. That one still does not disinfect. There's no disinfection there, but O'Brien and Calumet do. So I've done some work on looking at that and how the wastewater helped shape the river under normal, quote, normal conditions. The Chicago River, the flow of the Chicago River is about 70% wastewater, wastewater effluent. So it's mostly, most of the Chicago River now is coming from our wastewater treatment plants. And not only that, there's all these combined sewer overflows. So all these black dots are places where raw, untreated sewage can enter the river when it rains. So this has, allowed me to look at some of these these big ecological questions about microbial communities, but on a really practical, relevant, local, hyper-local scale here in Chicago. And we've done even more than just look at overall communities and how the wastewater influences them, but also this is a project with the Chicago Biomedical Consortium, looking at how, so when wastewater gets treated and even disinfected, the goal is to remove nutrients, mostly nitrogen related compounds, and to remove enteric bacteria like E. coli, like any fecal indicator bacteria. There's no treatment to remove chemicals of emerging concern. We see a lot of like caffeine, like things that people excrete. Caffeine, we see a lot of different drugs and drug metabolites. And certainly we see antibiotic resistance genes, especially at the height of like antibiotic resistance or antibacterial soaps and stuff like that. So people are washing with antibacterial soaps. All of that enters the river, and so we don't discharge into Lake Michigan, but there's about 100 wastewater treatment plants around between Illinois and Indiana and Michigan and Wisconsin that do discharge into Lake Michigan. And so we did a lot of work to look at antibiotic resistance genes and how they get disseminated in the environment and what those implications are to the ecosystem as a whole. And it's just some more pictures of what it looks like. A PhD student and postdoc working in Lake Michigan, also working at wastewater treatment plants, getting samples from wastewater treatment plants. Turns out wastewater treatment operators, plant operators are eager to help with science and give you samples or let you sample. So we've had some really great relationships with different facilities in the area, which has led us to this project that I'm working on now, that I like a lot of us, I think we kind of, when the pandemic hit a little bit over a year ago, many of us had to assess how we were going to do a lot of our work. Research vessels, ships weren't going to sea. They've started back recently, but they weren't going to sea. Our lab was closed initially at the beginning of this last year. And so we got this opportunity, like many across the world. I was going to say across the country, but this is really a worldwide effort to do what's called wastewater-based epidemiology. And sometimes people refer to it as wastewater surveillance. And this is a huge collaborative effort. We were funded by the Walder Foundation, which is a philanthropic foundation based in Chicago, based in Skokie, actually. I don't know if anybody's familiar with them, but they're the Walders, the family, are Orthodox Jews. When we met them, when we first got funded, it was a fun conversation. We also got funding from the Discovery Partners Institute, and we're working with Current, which is this water hub in the city. We're working really closely with the Chicago Department of Public Health, with the Metropolitan Water Reclamation District. We have a team of scientists. So I'm leading this group along with, we have people from Argonne and UIUC and University of Chicago, Northwestern. It's this huge effort. And the idea is that wastewater is really a composite biological sample if there ever was one. So if you get, if you've had nasal swab, if you've gotten a COVID test, that's a single individual, and that happens. A lot of individuals get tested. But if you look in wastewater, you're getting a sense of the entire community that's served by either a sewer or a wastewater treatment plant. So everyone contributes, whether they go to get a nasal swab or not. And this really took off when people first realized that the RNA from this virus, from the coronavirus, is shed in stool, and it's shed whether somebody is symptomatic or pre-symptomatic or asymptomatic. And so, but because of that, we can detect the RNA in wastewater, and we can get a better sense, a bigger picture of community infection than if we just looked at clinical data. And we're working really closely, I said, with public health officials. So we found, and this is consistent with what others have found across the world, that we can get information about changing levels of infection in a community over a week, from anywhere from 5 to 10 days before we start seeing those patterns in the clinical samples. So it's been, I have to say, this has been the most collaborative science I've done in my career. Everything, living on a ship and working with others really closely on very cool scientific projects. It's all been very collaborative, but in this case, but that's all like people who are funded together, working together. In this case, this is people who independently are doing this in their municipality, their city, their country, and where people keep using the metaphor of like building the plane as it's flying. Like, we are putting this together. It's moved so fast in the past year, and people are sharing their methods and talking to each other. We have a Slack group with thousands of people. We meet with the people who are doing this in the UK and Italy and the Netherlands and all over the US, like sharing, what kit did you buy? What method are you using? We're seeing this. How do you detect this? It's been so incredible and really collaborative. So just a little bit about what it entails. So I've gotten to apply all this wastewater work I've done and looking at microbial communities in wastewater to looking specifically at SARS-CoV-2 in wastewater. So we collect samples either from wastewater treatment plants or from sewers, and we extract the RNA from those samples, and then we run a PCR, which had I been asked to speak to a group like this a year ago, I wouldn't just throw out PCR. But now I feel like a lot of people know what PCR is because we talk a lot about it for like nasal swabs for COVID tests. So we do really close to the same thing that's happening with the nasal swabs we do for wastewater. And we can also do sequencing. So if you follow the news at all, you've heard about the variants and variants of concern and variants of interest. And so we can target those too, and we could look for different genotypes or types of virus in wastewater samples too. And we've gotten a lot of press for this over the past year. So this is my colleague Abhilasha Shrestha in the School of Public Health at UIC. This is my PhD student. This is Christopher Owen, a PhD student in my lab. And this was from a article that was in the Chicago Tribune. A couple months ago, we were on the news last month. Last March was last month, right? and last year and yesterday. So we were recently on local news. We've been working with the Cook County Jail, and this is a really cool relationship because we get samples twice weekly, and we talk directly to the infectious disease physician at the jail. And he will tell us that they think there's no cases. They do regular clinical testing, regular clinical sampling, and they ask us to sample this division or this unit based on what they're seeing or not seeing. And we've been able to detect cases before there's been a clinical test that clinical positive. So this is a really great relationship with the Cook County Sheriff's Office and the Chicago Department of Public Health. And now this is a lot of words, but the idea is that where we are now is developing these ideas that we're using with the Cook County Jail and with the local wastewater treatment plants and local sewers to really identify hot and cold spots in the city, to improve our precision and developing an effective public health response to what we see. We're still working on optimizing lab methods, but so is everybody else who's doing this. And looking for variants, which CDPH and the IDPH, the Illinois Department of Public Health, are really keen on. And what's really exciting is that we're about to embark on a statewide surveillance program that I'm leading, which is a little bit crazy, but in partnership with the Illinois Department of Public Health, we're developing a strategy to do this over the entire state. We're working to figure out which treatment plants to target. And what's key here is that I don't think I mentioned this at the beginning, but that big team that we have includes microbiologists. It includes public health experts and epidemiologists. It includes modelers. So this is a lot of the modeling work to find optimal locations and to figure out what to both improve the spatial precision, but also to figure out how to interpret and understand our results. Modelers, engineers, environmental engineers, yeah, really diverse team, ecologists. And just to end, we all kind of have tunnel vision for COVID-19 right now, understandably so. But the idea of wastewater surveillance or wastewater-based epidemiology is not new. There's evidence that this was done as early as the 1920s. A really famous case famous use of wastewater-based epidemiology in 2013 or 14. Wastewater-based epidemiology was used in Israel to detect polio before it became before there was an outbreak. So they managed to, but prior to that, they hadn't had a case of polio in decades, and so they were able to use the wastewater surveillance data to make sure that there wasn't another polio outbreak. And it's used not so polio, but it's used for all sorts of other infectious diseases, including influenza. And when we wrote some of these proposals, we talked about COVID-20 and COVID-21, and whatever's next, future pathogens that might be problem, we won't have to ramp up next time. Having this in place will be better poised to respond. The other thing that we can do is, and this directly ties into some of the other work that we're involved in, like looking at antibiotic resistance genes, we can look at not just diseases or pathogens, but we can look for chemicals or chemical things, chemicals of concern. So antibiotic resistance genes, or even antimicrobials. And our Chicago Department of Public Health partner is really interested in opioids. And this is a screenshot from the city of Tempe in Arizona, where they've been tracking opioids and other drug metabolites in wastewater. So our CPH partner really wants to be able to do that here as a way to, it's kind of the same way that our COVID-19 work, our findings can, we can go to public health agencies and say, you need to deploy mobile testing units or hospital beds, get ready for, a surge in cases in the hospitals or whatever. So being able to track opioids, there will allow the public health to respond to use that hopefully will keep people alive. So we just put in an NIH supplement to do this in Chicago, and hopefully we'll get selected, and we'll be sending our samples, our wastewater samples from here. And it all comes back to like ecosystem health and water quality and sustainability and the health of our environment. And I think, at least in my mind, understanding both pathogens and chemicals and the microbes that are there, the communities and how they interact, all ties together to be able to better understand our planet. So I'll end there, and I am happy to take questions.
Well, first of all, I want to say thank you. That was wonderful, an incredible amount of information, and just had me on the edge of my seat, which was great. I'm gonna start off with a question that came through quite a bit earlier in your talk when you were talking about how microbes work in the environment. And there's a question from Kay Greenfield about discovering microbes to actually do the job that we need to consume and break down plastic waste in the oceans and across the earth.
We have, yeah. So there are microbes that are able to eat there are microbes that are able to eat lots of things. So Frank asked at the very beginning what my favorite microbe is, and one of the coolest things, I didn't have a good answer, but one of the coolest things about microbes is they eat everything, like not an individual, but there's microbes that can literally breathe rocks, like breathe solid rock. And so I think it's not a matter of finding or coming up with this idea that maybe there's a microbe that can do this. It's finding the microbe that can do that. We know that they probably exist, but in this case, we know that there are microbes that are able to eat plastic, that they can degrade it. It's just a matter of like deploying it. If you're thinking like large scale, like the plastics in the ocean, it's to be able to do that on a large scale. That's where the technological application gets really complicated. But they exist, and I think our future is microbial. Like we're going to need to depend on these organisms to do a lot of the things that we've messed up, or to fix a lot of the things that we've messed up.
Well, at least we have some tools, I guess, right?
Yeah.
So while you were talking about the wastewater based epidemiology, I had put a question in which you sort of answered about what other organisms we're looking at, but I wondered if you could just say a little bit about the extent of the numbers of different diseases and things that we're looking for constantly through the wastewater study.
So we're there is no number. The cities that are doing routine monitoring are probably monitoring for maybe 5 or so different maybe 10 different pathogens at a time. And actually there's like the CDC has like kits or like a set of organisms that you can look for in wastewater. In this case, right now in Chicago, we're only looking at the coronavirus. We have, but because we're a little bit late to the game, so we started, everything just moves simultaneously slow and fast. But we set this up, we really got going in September, October, in the fall. And now we're moving to a statewide effort that's going to be up and running in a couple weeks. And at this point, a lot of cases, although they're starting to climb again, but they're much lower than they were at some of the earlier peaks. And so one of the things that we're doing is setting this up so that we can monitor other things. So we kind of are keeping the long vision, like what can we look for in the future?
Yeah.
And what is going to be meaningful for public health agencies and for our health.
Well, I suppose as your familiarity with the testing techniques gets better, then when something new comes up, probably you can design the test a lot faster and deploy it more quickly, right?
Totally. Yeah.
Okay.
Exactly. So
Well,
if we knew something was coming or something was there, or like even to do this annually for influenza, like or to be able to see what's in the environment and be better able to both treat and prevent something that's common.
right. Thank you. Ken Heiferman asks about antimicrobial substances that we have, prevalent in our environment right now, and what the bigger picture is for the danger to society, because it somehow forces the selection of certain things over others.
Yes. No, it's a huge problem. And actually, if you talk to a lot of people in public health right now, they will tell you that they thought the next huge public health disaster, until COVID hit, they were really expecting the next big public health disaster to be related to antimicrobial resistance. This is still a huge problem. Overuse of antibiotics, and not just in medicine, like having over prescribing antibiotics, but also antibiotics added to everything. There's a period of time there before the FDA started clamping down on that, where there was like triclosan and toothpaste, like it was everywhere. You could get antibiotic everything. And yeah, right. And it was such a problem, and it still is a problem that people are getting sick with things that are antibiotics that are available in medicine can't treat. And I have a number of colleagues, including one whose picture was on one of my slides, who's in the College of Pharmacy, where he's really, his research focus is discovering new antimicrobials, so that we have this like new bunch of tools that we can dip into when nothing else we have works. And from my perspective, the release of all this stuff into the environment helps exacerbate that, because it can then be spread. Bacteria and archaea both are really notorious for exchanging genes between each other, and the genes where antibiotic resistance is, those are really mobile. So that's why it's such a problem, because they can very easily make their neighbor resistant and spread it really fast. So the question was, do I think it's can be more dangerous to society? 100%. And I think that wastewater treatment plants haven't had it on their radar as something that they need to, or even not just the plants themselves, that plants follow EPA guidelines for treating our wastewater. But I think we really need to start thinking about treating our wastewater not just for the EPA standards, but adding to that antibiotics and other chemicals that are having an effect on the environment and on us.
So would it be even possible on a big scale to filter all of those things out of the effluent?
So I had a paper a year or two ago about disinfection. So once disinfection was implemented, like how that affects other genes and chemicals. And I think there are ways there are ways to wastewater treatment is it's a big deal field for like a big industry, and there's and it's pretty high tech now. And I think there's a lot of ways that we can implement additional steps to, whether it's filter based or UV treatment or whatever else. But we actually, so one of the really interesting things with this wastewater COVID stuff is that we see SARS-CoV-2 RNA in the effluent, in the stuff that comes out of the wastewater treatment plants, not just in the raw sewage, which we would expect because that's like feces, but we see the RNA still in what comes out. And so we don't know if that's a problem. To Kenneth's question, like we don't know if the RNA is, I don't think it's virus, and I don't think it's viable virus, but if that RNA can be a problem. So I think, you know, it's not I'm an environmental engineer by association, but there's a lot of people who are professionals in this area. But I think it's something that we collectively need to think about, is getting rid of this stuff, because it all goes into, you know, we don't discharge into Lake Michigan anymore because the Chicago River, the Army Corps of Engineers reversed the flow after people were dropping dead of cholera. But it does eventually flow and meet up with other tributaries and ends up in the Gulf of Mexico. And so we're not drinking it, but it has.
Somebody else is.
Right. So
if anybody has a question they'd like to ask directly, please feel free to unmute yourself and ask away.
Rachel, what is the technique that's used by wastewater plants to disinfect effluent?
Oh, you want to get all technical, huh? There's actually a variety. So when they implemented it here in Chicago, O'Brien they built a system to do UV disinfection. So they just blast the treated wastewater with UV light for a set of EPA regulated amount of time and then discharge it. The one in Calumet, they do a combination chlorination, dechlorination. Some facilities do both. Some do varieties of those. Interestingly, here in the Midwest, most wastewater treatment plants that do disinfect turn off disinfection over the winter because they're like, nobody's recreating in the water in the winter, so we don't need to do it. So here in Chicago, they disinfect they turn it back on last month. So March till about October is when they disinfect, and then they turn it off for the winter. And that's true across the Midwest.
But UVA is ultraviolet.
Yes, sorry, ultraviolet light, yes.
Well, if nobody else has a question, I've got one more. I just saw a very interesting article online from National Geographic about these new super giant viruses that they have found that have all kinds of stuff in them that looks a lot more like microbes as opposed to a virus, and also giving them a chance to possibly connect viruses to what they call living cells, right? And I just wondered if you had any insights about that research and what's on the horizon.
Oh, those are so cool. They're mini viruses, and they're giant viruses. Yeah. In terms of you know, I feel like with all the new discoveries about viruses, they're really blurring the lines between living and not living. A lot of the virus, I told you that like there's about 10 times as many viruses in the ocean as there are bacteria.
Yeah.
And most of those viruses infect bacteria. So that's their host. Some viruses infect other viruses. And there's all these, so the giant viruses do this, but there's also some smaller viruses that carry things like genes for photosynthesis. And when they infect a bacteria, they give the bacteria the ability to use light to photosynthesize. And so it's almost like they're not, they're living things sort of, but they're also providing the ability for living things to be the living things that they are. And when I showed that crazy tree with the bacteria, the archaea, and the eukaryotes, it's such interesting evolutionary questions. We think that eukaryotes, that we and plants and fungi are our closest relatives, arose from a combination of an archaea and a bacteria, possibly, or an archaea, and so an endosymbiosis, like they kind of join together. So there's this really interesting question about not just like the origins of life, but where life is going with these viruses that can kind of make new organisms almost. It almost gets philosophical, right? Yeah. It's a good question.
It was just fascinating stuff. It made me realize how little I paid attention in biology and chemistry class. Even though I went to class and got good grades, I just didn't absorb enough of the big picture ideas that you're able to share with us, which is really wonderful.
Well, I'll tell you, my mom likes to say that things weren't invented when she was in school. When I tell her about things, she's like, oh, that wasn't invented yet. But actually, I will tell you that it actually holds for archaea. So archaea were discovered in 1977. So if you were, not judging anybody's age, but if you were in school prior to that, you probably learned about the 5 kingdoms, which is not true anymore. But that's something that we've only known for a couple of decades now. So there's stuff that even if you did pay attention, Charles, it might be different now.
Right, right. Well, if there are no more questions, I want to say thank you very much to Rachel for a wonderful, wonderful Temple Talk, and
Pleasure.
Thank you for joining us.
No, fascinating. Thank you.
Thank you.
Yes, thank you very much.
And of course,
Rachel.
If you want to see any of this again, it will be on our Facebook page, and it will soon be up on our YouTube page. Feel free to share the links with anybody that you think will be interested. The board member, Peter Nordlander, who analyzes all of our social media hits for people who are viewing what we've got available, said that in addition to the people who've come to the Temple Talks, there have been over 250 people looking at those videos from outside the group. And so that's really wonderful that we're creating a small body of work here that people are coming to look for. And so spread the word, send it to your friends and say, join us next time. And then the next time,
Charles?
Yes.
What link would we give to somebody to look for example, today's lecture? How would I tell someone to?
Well, the fastest way, Frank, would be to go to our Facebook page, because this was live streamed on Facebook. So there will be a video there almost immediately. And if you right click on the video itself, you can copy the URL and you can send that by email to anybody you want. On the YouTube channel, it'll take us a couple of days to move the video over and get it archived. But the Facebook page is almost immediate. So as soon as we're done here, if you wait about 5 minutes, that video will be ready to go, and you can give them the link.
What title would you look for under YouTube?
So on YouTube, when you go to our Temple Har Zion channel, we actually have some archived menus, and one of them says Temple Talks. So
Okay.
We're hoping that it's fairly self-explanatory. We are in the process of revamping our social media presence, and this is an ongoing process that's going to take another couple months to complete. But hopefully we're going to have a nice little catalog of videos and links to the things that we offer that will make it very simple for people to come visit us. Well, thank you very much, Rachel. And it was really a wonderful talk. And I'm so glad that I stopped you in the parking lot and asked you if you would join us.
My pleasure.
All right.
Great.
And for everybody else, I'm going to say good afternoon and enjoy the rest of your weekend.